Hello everyone, I welcome you all in my Science Academy YouTube channel. So today is lecture number 7 of biochemistry, and in today's session we are going to deal with the topic enzyme. I have divided enzyme in 4 different lectures will be there for enzyme. In first lecture I will talk about introduction about enzyme, and second classification of enzyme, and I will talk about history of enzyme. These 3 topics I will talk about in today's session. So let us start with the introduction about enzyme. And what is enzyme? Enzyme are protein, generally it is a protein which selectively convert substrate into product of any metabolic pathway. That is called enzyme. So when substrate is there, in presence of enzyme, this will convert into product. This is most of the time protein, but there is an exception. Ribozyme or RNA is also enzyme, but it is not protein. So all enzyme are not protein. Discovery of enzyme. In 1700, people have analyzed that complex food convert into simple food by process of digestion. So how it is happening? Some molecule is converting basically complex molecule into simple form. 1800, people have analyzed that saliva or plant extract can convert carbohydrate into sugar. In 1800, around 30s, Louis Pasteur first analyzed that sugar can be converted into alcohol by the process of fermentation. The organism which actually converting sugar into alcohol, it was yeast. In 1896, Edward Buckner observed that the molecule which is actually converting sugar to alcohol conversion is present inside the yeast. So whole yeast is not required. If we are going to take yeast extract, then it can convert sugar into alcohol. So first time it was observed that the whole organism is not required to convert sugar into alcohol or ethanol. There is a molecule, certain molecules, biomolecules are present in the yeast which can be extracted, which can be separated, and that can convert sugar into alcohol. So biomolecule can be separable. Initially, before Edward Buckner, it was assumed that this molecule cannot be separable. This is present in the organism, and that's why we have to use organism for the conversion. Now, F.W. Kuhne was scientist who named this biomolecule as enzyme, and later on in 1926, Sumner was first scientist who analyzed urease enzyme, that which is made up of amino acid. Now we are going to move about enzyme classification. 6 class is there, and first is called oxidoreductase, second is known as transferase, third is known as hydrolase, fourth is known as lysase, fifth is known as isomerase, and sixth known as ligase. So how we can remember this? OTHLIN. Now coming to this, oxidoreductase are those enzymes which do reduction and oxidation. This is most common. Which do the chemical reaction which convert substrate into product by the process of reduction, addition of electron or removal of electron, either reduction or oxidation process. Transferase, which transfer functional group from one molecule to another molecule. So it is transferring the functional group like OH from X to Y. Suppose it is X, OH, when reacting with Y. So reacting with Y. So now it will convert X plus Y and OH. So OH will come here. So this transfer over here is known as transferase. Hydrolase, removal of functional group that leads to water formation. Suppose this is a X and there is a one function group OH, then this is combining with another H, so what will be the outcome? X and Y with the removal of, so hydrolase will always carry the reaction where the water molecule can be formed. So this OH and this will combine together to form water. Now next one is lysis. Here conversion of double bond to single bond by addition of functional group or vice versa. Suppose this 2 carbon is double bond and this is attached with CH and CH. So here if suppose functional group like H2O has come, then it will form C single C H H. Now OH will attach over here and this converted into single bond or vice versa. This is known as lysis. So conversion of double bond to single bond or single bond to double bond by the addition of certain functional group is known as lysis. Now next one is isomerase. Isomerase is transfer of functional group from one carbon to another carbon of same molecule is known as isomerase. Suppose C double OH NH3 H CH2 CH and CH3 CH3 like that is leucine. And if this carbon will goes in this position, second, third position, first, second, third, fourth, so this carbon will go to the third position and H will come to the fourth position, then it will be isomerase. So how it will looks like C double OH NH3 H CH, this carbon will come over here and this is become CH2 CH3. So isomers are same molecular formula or chemical formula, same chemical formula but different structural formula. These two are different in structure. Next one is ligase. Does condensation reaction means addition reaction, you can say, combine two different molecule to form a single complex molecule by use of energy in the form of ATP. So this reaction always required energy. This is a very important thing. Rest all 1 to 5th class doesn't require any source of energy except 6th one ligase, which always work with the help of energy. So this is about the enzyme classification and this is in the fixed order. You cannot change first with the second or second with the third one. So we are discussing with enzyme commission number, ECC number. So each enzyme has unique number. Let us take example like ATP plus glucose. It will form glucose 6-phosphate plus ADP. This reaction is done by enzyme known as hexokinase, common name, and scientific name is glucose ATP glucose phosphotransferase. And enzyme commission number for this is 2711. Now you have to understand what is a 2. 2 is always showing class of enzyme. So we have seen second class was transferase. Now second is subclass, like which type of functional group it is transferring, alcohol, phosphorus or H, methyl group. So this is about 7th class. Seventh is subclass. Third one, which is first, is acceptor atom of functional group, like in sugar molecule CH2OH. So this is hydroxyl group is a acceptor of phosphate because phosphate will add over here. So OH in this case is a acceptor. Next one, so acceptor molecule, which is in this case is glucose. So this is about first is class, second is subclass, third is a acceptor functional atom, and fourth is an acceptor molecule. Now we are talking about characteristics of certain enzymes. Some enzyme work alone, that doesn't require any help or any other functional group or any other cofactors. That is called apoenzyme. But some enzyme requires non-protein molecule, then it is called holoenzyme. This enzyme can only work when non-protein molecule can be associated with this enzyme, and this non-molecule enzyme can be cofactor or coenzyme. Cofactor are generally metal ions. Coenzyme are organic molecules like here iron, copper, zinc, nickel, cobalt. Here organic molecule like NADH, lipoic acid, FADH2, coenzyme A. These are the organic molecules. Heme is another complex. So this organic molecule. So these are coenzyme. Now another term is that cofactor or coenzyme can associate tightly to protein by covalent bond. If it is like that, then it is called prosthetic group. So prosthetic group, cofactor and coenzyme should be very clear. This is a part of non-protein molecule that's called combined with the enzyme to form holoenzyme. There are certain examples like cytochrome oxidase. I will write one example which requires either copper or iron. Then dinitrogenase, another example, which requires molybdenum. Alcohol dehydrogenase, which requires zinc. So these are the certain examples which only function in presence of this cofactor or coenzyme. Hemoglobin, which requires heme, so that's a coenzyme. So this is about the introductory lecture about enzyme. In the next lecture we are going to discuss with the how enzyme works. So that will be the second lecture of enzyme. We have divided enzyme into 4 lectures. Today was the introduction lecture. Second lecture will be how enzyme works. Hope this video is going to be helpful for you in understanding the interesting topic which is enzyme. Please do subscribe my channel. Also share this video with your friends. And please hit the like button and also comment in comment section that whether you have liked this video or not. Thanks a lot for watching.